Hello students, today we will be seeing Histology of GIT, Large Intestine, Part 2. In the video of Large Intestine Part 1, we had seen the general features of histology and gross anatomy of large intestine, and we studied one important slide, and that was of histology of colon. In the video today, we will be studying the other important slide of large intestine, that is histology of vermiform appendix, and we will then be seeing how to differentiate between small intestine and large intestine. Now, vermiform appendix. This is a blind-ended tubular appendage extending from the cecum. It is the narrowest part of the gastrointestinal tract. Morphologically, it shows three parts: base, body, and tip. Histology of vermiform appendix. A transverse section of vermiform appendix shows four layers from within outwards: mucosa, submucosa, muscularis externa, and serosa. Mucosa. This consists of three parts: the lining epithelium, lamina propria, and muscularis mucosae. Mucosa is lined by simple columnar epithelium with microvilli, numerous goblet cells, and M cells. Lamina propria contains short tubular intestinal glands or crypts of Liberkuhn. Glands are lined by simple columnar cells with numerous goblet cells. Lamina propria is mostly occupied by large and small lymphatic follicles or nodules that may also cross the muscularis mucosae and extend into the submucosa. This is the characteristic feature of histology of vermiform appendix. Now, what we see here is a diagram showing us the transverse section of the vermiform appendix. The entire transverse section is seen here. This is the luminal surface. So, when we go from here outwards, what we see here is the mucosa, then is the submucosa, the muscularis externa, and the serosa. Now, what we see here are the small and large lymphoid nodules or aggregations which are seen in the lamina propria. They are seen all along the circumference of the wall of the vermiform appendix, and at places it may also cross the muscularis mucosae and enter into the submucosa. So this entire circle of lymphatic particles is the characteristic feature of transverse section of vermiform appendix. And then what we see here is a magnified view of a part of the section, where here we appreciate that this is the mucosa, the submucosa, muscularis externa, and serosa. So mucosa here shows the lining epithelium, the lamina propria, and the muscularis mucosae. So lining epithelium shows simple columnar epithelium with goblet cells. The number of goblet cells are much more than what are seen in the previous slides of small intestine as well as large intestine, which we have already studied. So simple columnar epithelium with numerous goblet cells. Lamina propria showing crypts of Liberkuhn and the lymphoid nodules. Muscularis mucosae shows smooth muscle fibers arranged as inner circular and outer longitudinal layers. Submucosa consists of dense irregular connective tissue. The same diagram here showing us the muscularis mucosae, which is seen as inner circular and outer longitudinal layers of smooth muscle fibers. These are sparse, thin layers that are seen. And then we see the submucosa, which is made up of dense connective tissue, showing blood vessels and adipocytes. And at places, we also see some lymphoid aggregates which have passed or crossed the muscularis mucosae and have entered the submucosa. Next, we see the muscularis externa. This consists of a thick layer of inner circular and outer longitudinally arranged smooth muscle fibers. Again here we need to note that appendix does not have tinea coli. And finally we see the serosa. Appendix is entirely covered by peritoneum, that is mesothelium, and hence the outermost layer is called as serosa. So what we see here is the muscularis externa showing inner circular and outer longitudinally arranged smooth muscle fibers, and outermost is the serosa, which is connective tissue showing blood vessels, adipocytes, few lymphatics and nerve endings. Now what we see here is a scanner view or a low power view of an H&E stained slide of vermiform appendix. What we see here are mucosal folds. This is the mucosa. We see lining epithelium and we see the lymphoid aggregates which are seen all along the circumference of the lamina propria. Then we see a thin muscularis mucosae, submucosa, muscularis externa, two layers, inner circular and outer longitudinal layer, and finally we see the serosa. Another low power view showing again the mucosal folds, the mucosa showing lining epithelium, the crypts of Lieberkühn and the lymphoid aggregates which are seen all along the circumference of the wall in the lamina propria, the characteristic feature of histology of appendix. A high power view showing us the luminal surface here, the lining epithelium, the intestinal glands or crypts of Lieberkühn, lymphoid aggregates, a thin muscularis mucosae, submucosa and part of the muscularis externa. An aperio image scope picture. What we see here is the entire transverse section. Please appreciate the complete circle of lymphoid aggregates. And this here is a magnified view showing us the lining epithelium, lower part showing the crypts of Lieberkühn, lymphoid aggregates in the lamina propria, a very thin muscularis mucosae, submucosa, muscularis externa and serosa. A magnified view of the lining epithelium here shows us the number of goblet cells that are seen in between the simple columnar epithelial cells. The salient features for spotting for a slide of histology of vermiform appendix transverse section will be presence of lamina propria containing lymphoid aggregates, presence of thick muscular layer in the muscularis externa, and absence of villi. Now that we have studied all the slides of histology of small intestine, that is the 3 slides which we see there, duodenum, jejunum and ileum, and the 2 slides in histology of large intestine, that is histology of colon and vermiform appendix. Now let us see how do we differentiate between firstly the 2 slides of large intestine. How do we differentiate between histology of colon and histology of vermiform appendix? Now when we look at the lymphatic nodules, in colon a solitary nodule may be seen in the submucosa, and this is true for any part of the GIT where we can see a solitary nodule in the submucosa. Whereas in vermiform appendix we see large and small lymphatic follicles which are seen all along the lamina propria, all along the circumference of the long of the wall of the appendix, and it may cross the muscularis mucosae and extend also into the submucosa. So a complete circle of lymphatic follicles in the lamina propria is a characteristic feature of the vermiform appendix, while in colon it may be just a solitary nodule which is seen in the submucosa. When we look at the characteristic features of the large intestine, like tinea coli, circulations and appendices epiploicae, all these three are seen in the colon and they are absent in the vermiform appendix. Now let us see these differences in the diagrams. Here is histology of colon and this is histology of the vermiform appendix. Now as we see here, a solitary lymphatic nodule may be present in the lower part of the lamina propria or in the submucosa, whereas in vermiform appendix we see an entire circle of lymphatic aggregates which are seen all along the circumference of the lamina propria and may also cross the muscularis mucosae and enter into the submucosa. Whereas in the muscularis externa, we see tinea coli, which is a thickened band seen in the longitudinally arranged smooth muscle fibers, which is absent in the vermiform appendix. So that is how we differentiate between the histology of colon and histology of appendix. So two major features, the solitary nodule which may be seen in colon, whereas the complete ring of lymphoid aggregates in the lamina propria which are seen in the appendix, and the tinea coli which are present in the colon and they are absent in the appendix. The low part view of these two, this is a slide of colon and this is a slide of vermiform appendix. So as is seen here, in the colon we hardly see any lymphoid aggregate, whereas here in the appendix we see the entire ring of lymphoid aggregates all along the circumference of the wall in the lamina propria. And what we see here is a tinea coli which is absent in the vermiform appendix. Next, let us see how do we differentiate between histology of small intestine and histology of large intestine. Now, when we look at villi, these are characteristic features and they are present in the small intestine and they are absent in the large intestine. When we talk of mucosal folds, these are absent in the small intestine and they are present in the large intestine. When we look at the crypts of Vermiform, these are very few and are seen in the lamina propria and are less deep in the small intestine, while they are numerous in the lamina propria in the large intestine and are much deeper as compared to those that are seen in the small intestine. When we talk of the number of goblet cells, these are less in number in the small intestine and these are predominantly seen in the lining epithelium in the large intestine. When we talk of the longitudinally arranged coat of smooth muscle fibers in the muscularis externa, this is uniformly thick in the small intestine, whereas it is interrupted to form tinea coli in some parts of the large intestine like the colon. This here is a diagram of histology of ileum, part of small intestine, and this is histology of colon, part of large intestine. So let us see the differences here. What we see here are the villi present in small intestine and what we see here are mucosal folds which are present in the large intestine. And second most important is what we see in the muscularis externa, where the longitudinal layer, outer longitudinal layer of smooth muscle fibers is uniformly thick, while here we see the tinea coli, which is a characteristic feature of the colon. What we see here are the low part field views of the H&E stain slides of the ileum and the colon. So in the ileum, what we see here is the villi which are seen on the luminal surface, whereas what we see here are mucosal folds. And the second feature which we see here is that the muscularis externa, the longitudinally arranged fibers of smooth muscle are uniformly thick, whereas we see here the presence of tinea coli. Next we see the difference between histology of ileum and Histology of Vermiform Appendix. Since both these slides have lymphoid aggregations in the lamina propria, how do we differentiate between the two? Now, when we talk of villi, these are projections of lamina propria. The core of the villus contains blood vessels and lacteals, that is lymphatic vessels, and these are much smaller as compared to the mucosal folds. So these villi are present in the ileum. They are a characteristic feature, while they are absent in vermiform appendix. Whereas mucosal folds, these contain a core of lamina propria and are much larger as compared to villi. These are absent in ileum and they are a characteristic feature of vermiform appendix. And most important, lymphoid aggregates or lymphoid nodules. These are seen as Pierce patches in the lamina propria in the ileum, and they are found only along the anti-mesenteric border of the ileum. So they are not seen in the entire circumference, but they are seen only along the anti-mesenteric border. Whereas in vermiform appendix, these lymphoid aggregates are seen along the entire circumference of the wall of the appendix in the lamina propria and may at times cross the muscularis mucosae and also enter into the submucosa. Now what we see here are the diagrams of the ileum and the vermiform appendix. So villi are characteristic features in the ileum, whereas mucosal folds are characteristic feature of the appendix. When we talk of lymphoid aggregates in the lamina propria, these are seen only along the anti-mesenteric border in the ileum, while in vermiform appendix, they are seen all along the circumference of the wall in the lamina propria. So these are the characteristic features which help us to differentiate between a slide of ileum and a slide of vermiform appendix. Thus, we see the low-far views of the H&E stain slides of ileum and of appendix. Now what we see here are the villi which are seen along the luminal surface, and what we see here are the mucosal folds. While lymphoid aggregates which are seen in the lamina propria here only along the anti-mesenteric border, whereas in the appendix they are seen all along the circumference of the wall in the lamina propria. Thus we have studied the important slide of histology of vermiform appendix in this video, and finally we also saw how to differentiate between the histology of small intestine and large intestine, how to differentiate between the two slides of large intestine, that is colon and vermiform appendix, and also how to differentiate between a slide of ileum and vermiform appendix. For further reference, you can read Sontakke Yogesh, Textbook of Human Histology, CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 219 to 220. Thank you.